Excellent. All right. So welcome, everybody. Before I introduce our speaker tonight, I'll announce the date of our next speaker. So make sure you make a note, October the 17th, and I'll also put it in the chat, when Dr. Elizabeth Bowman, a fungal ecologist, will speak to us about the impact of climate change and related disturbances on soil microbial communities. So get that date on your calendar. It's going to be really interesting, October 17th. Tonight, we are happy to have Dr. Brian Wilsey with us to share his knowledge and his research on the impact of climate change on native tallgrass prairies. He is a professor at Iowa State University in Ames, Iowa. He has studied grassland ecology for the past 25 years on different topics like the impact of climate change on interactions between plants and grazers, the relationships between biodiversity and ecosystem functioning, species diversity maintenance, carbon and nutrient cycling, and community assembly. He has visited and worked in grassland systems around the world, including the tall and midgrass prairies of central United States, the savanna grasslands in Tanzania, the steppe in China, and the flooding pampa of Argentina. He's published widely on these and other topics. He is currently doing some research to really quantify the impact of pollinators on species diversity of forbs, and actually being able to quantify how, when pollinators are eliminated, the species diversity decreases and the life span of a particular forb gets much shorter. And interesting about him, his wife is a historical mystery writer, and so they travel a lot for her research, and they just got back from Scotland. So all of that is to say, welcome, Dr. Wilsey. Thank you for being with us, and I will turn this over to you.
Okay, thank you.
And let me also say, before Dr. Wilsey gets started, we're pretty sure we're going to have a little bit of time at the end for questions. And so if you'd like to put them in the chat during Dr. Wilsey's presentation, that's great. Or if you want to just ask them at the end, you can just raise your hand or unmute yourself and ask the question. So we will try to leave time for that.
Okay, thank you. I hope everybody can hear me. And let me move at the slideshow. So I'll try to make this as entertaining as I can since it's after 5 o'clock. But I'll talk a little bit about the work that I've done in Texas over the years, especially. I'm kind of a semi-Texan. I lived in Stephenville, and I lived in Wichita Falls when I was a teenager. And then later I lived in Temple and Commerce. And for 12 years, from Ames here, I went back to Texas twice a year to work on a project that I'll tell you about. And so anyways, as I'll go through, I'll talk mainly about the impact of climate change on tallgrass prairies. So as an outline, I just want to talk about global change. I'm going to talk about different aspects of global change. So CO2 is increasing in our atmosphere, and that can have direct effects on plants and ecosystems. Of course, warming, land use change, invasive species are so widespread, and at least in grasslands, people think of that as a global change. And then altered precipitation. So rainfall is becoming more variable. We've done work on the rainfall variability, and it's actually increasing in some places, and for example, in Iowa. And I'm going to try to stress the results from the work that I did over the years with Wayne Pauley at the Grassland Soil and Water Research Lab in Temple, which is a USDA ARS lab. And before I get started, I just want to point out, I wrote the book on the biology of grasslands. So that's a joke. I like to make that joke, but because I did write this book a few years ago. And so some of it will come from the review from this book. But grasslands cover about 25% of the Earth's surface. And, they're called prairie, which is French for meadow. The pampas in South America. In China, I visited a lot of prairie-like systems that they call meadows. And then they have steppes, which have a little bit of a shrub component. And there's been a lot of reviews that I went through when I wrote this book. But one thing that's really interesting about grasslands is they're the most altered by humans and they're the least protected. So as a result of all that, a lot of us, and myself included, were really interested in restoring these prairie ecosystems and bringing them back. And in the Midwest, where I am now, restorations are really big, and the amount of acreage and restorations is a lot higher than the acreage in remnant prairie. So now a lot of it is not just protecting remnant prairie, but improving these restorations, which I'll talk about today. So before I get started on the numbers and the figures and everything like that, I'll just show you a few pictures of some grasslands that I worked in. And this is the Serengeti. I did my PhD in Tanzania. And this is called a kopje. It's Kopjes, but they say kopje. And this is a granite outcrop in the Serengeti Plains. So a lot of the Serengeti is just wide open grassland. It's not even savanna, and some of it's savanna, like this picture here. And I was always struck when I was in Tanzania how much it looked like Texas. So Texas, Tanzania, they really look similar. Here's a picture of the Asian steppe. This is Delhi Wang in China. This is a huge, huge grassland that extends all through a lot of Asia. Of course, in Europe, they have some human-derived grasslands, and they have some climatic grasslands as well. They're usually called chalk grasslands. And then, of course, in the Midwest, we have the Great Plains. So I'm mostly going to talk about tall grass prairie today, but if you go west, you hit the mixed grass prairie and then short grass prairie and steppe. So this is a picture that I took at Theodore Roosevelt National Park in North Dakota, and it's a bison feeding on a prairie dog town. They preferentially feed on prairie dog towns. And in a former life, I studied the effects of grazing a lot. And so I'm not going to talk about that very much today, but I have a lot of research on the effects of grazing. So anyways, in the Midwest, restorations are really common, and we're trying to improve it. And Nathan Soley, I notice, is one of the participants. And he and I have been working a lot on restorations and especially on plant pollinator interactions. He's an expert on pollinators. So if you look at where tallgrass prairies occur, you've probably all seen these kind of pictures. They extend from Manitoba to central Texas, and most people include the coastal grasslands of Texas as part of this. And I always look at the maps in Southwest Louisiana. It's a lot of that is prairie. And so if it's an accurate map, it would have Southwest Louisiana as prairie. And on the right is the prairie that's intact, that's left. And so it's mainly in the Flint Hills in northeastern Oklahoma, but tallgrass prairie exists as little postage stamps in other places. And what a lot of sites that used to be tallgrass prairie are now have kind of reassembled into exotic fields. So on the bottom, I have on the left a native prairie with some orchids and really diverse compass plants. And on the right is an exotic field. This is in the near Temple, Texas. And this picture I took near one of our sites, and this is King's Ranch bluestem Bothrochloa scamum. And this was not planted or not seeded anywhere. It just showed up. at one point, and it's spread everywhere. And basically, it's the dominant grass in a lot of the areas around Temple. So I've done a lot of research on the effects of this invasion compared to the original native prairie. So I'm going to go over different global change effects or climate change effects. And so the first topic that I'd like to talk about is just the direct effects of elevated CO2. So CO2 is rising in our atmosphere, and a lot of people talk about how that's going to affect temperatures, but it can also have direct effects on plants and grazing mammals. And during my early part of my research career, I looked at how elevated CO2 would affect plant grazer interactions. And what we consistently find over and over and over is if you put plants under high CO2, the nitrogen content of the tissues goes down. And another way of saying this is the protein content of the tissues goes down. And I always like to think of plant tissues as kind of like rice and beans in the human diet. A lot of people eat rice and beans, and it's kind of balanced. The beans provide the protein and rice provides the carbohydrates. Elevated CO2 kind of increases the rice component. So it's more carbohydrates, but lower in protein. And so that's what happens consistently when you put plants under high CO2. And it's been shown in field sites, for example, at Kanza Prairie, where they have long-term records. We know that the tissue concentration of nitrogen is going down over time. So that's a direct effect of elevated CO2. And Wayne Pauley, the person in the picture on the last slide, has done a lot of research on the effects of elevated CO2 on plants and ecosystems. And this slide is from some of his research. And what he found, he's been kind of uniquely studying CO2 at subambient through current levels for future high levels. And so, for example, in this picture on the x-axis is CO2 concentration. And I put an arrow about what the CO2 concentration is now. And if you go to the left of that, it's subambient. In pre-industrial revolution, values are about 270 parts per million, and they're rising through the present day, and they're expected to go up to about 500 parts per million. And if you look at the sandy loam here, that it's interacting with soil type, but if you look at the sandy loam, productivity is going up currently at the current levels of CO2, and it's expected to continue to rise in the future. Whereas the clay soil, it's expected to saturate. So it's kind of like a saturating function. And then the bottom panels are the fraction of Indian grass. So this is Sargasterum nutans, which is a really tall grass. And what he found in his studies is under high CO2, the fraction of Indian grass goes up. And that's a very productive tall grass. And so that indirectly affects primary productivity. So primary productivity goes up because of CO2, and it goes up because of this indirect effect of having more Indian grass. So that's really interesting. And I'm going to cover several results from an Iowa climate assessment that I was involved with. And I talked about Iowa in this report, but I was really talking about the tallgrass prairie region in general. And so I'm going to go over several results from that report that I wrote from a review. And several things that were really interesting in this review is one is elevated CO2 really favors wind pollinated species over animal pollinated species. And this is Louis Ziska's work on the right. And this is pollen production from ragweed that causes hay fever. And it really responds to elevated CO2. So if you just look at the top panel, it's pollen production from ragweed, which causes hay fever. And that's expected to really rise in the future. So it has 1890, the year 2000, and 2050. And we're going to have more problems with hay fever, I think, in the future. And then I've done a lot of work with invasive species. Invasive species, at least in grasslands, is viewed as a global change issue. Grasslands worldwide have some component of non-native species, and sometimes it's substantial. So this is a remnant prairie in November, and it's being invaded by crown vetch, Perennia varia, and you can see this crown vetch is actively growing when all the native species are dormant, and it's really spreading rapidly. So we've done a lot of work with invasive species or non-native plant species. And one project that I did that I was really excited about was with a student named Leanne Martin. And in this study, we sampled a gradient of sites all the way from Minnesota to central Texas. And I have the map here, and the little triangle is Temple. So we did experiments there. And as part of this study, we sampled a bunch of different sites, and they were paired. So they were either dominated by native prairie species or some kind of non-native or exotic species. And so they're all paired by latitude. And at each site, we showed up and sampled the plant community, and then Leanne sampled the bee community. And so we looked at, how invaded are they? And then how did plant diversity, how was it different between exotic sites and native sites, and so on. And just before we get to the data, so the exotic site, it turned out, was about 90% exotic biomass and 10% native. And the native sites were about 92% native species and 8% exotic. And so here are the results from this study, but this looks at a whole bunch of different measures: plant species richness, plant species diversity, bee abundance, bee diversity, bee species richness, and so on. And if the bars are negative, that means the native sites are higher than the exotic sites. And we found really large difference. The native sites had much higher plant species richness and much higher plant species diversity, and they had a greater bee abundance as well. And that part of this study, we looked at phenology, and we found that these invasive plant species are really altering phenology. So this is NDVI, which is measured from satellites, Landsat satellites that fly over the site and collect data on reflectance. And so we had sampled all of these sites so we could use spatial data from satellites to look at when things green up and when they senesce. And I kind of summarized the whole study in this slide, but the native sites are the sort of the normal curve, normal distributed bell-shaped curve, and the exotic sites are the dashed line. And basically the exotic sites had earlier green up and later senescence. And then in the summer, they had this dip. They kind of turned brown a lot of times in the summer. And we saw this pretty much across the gradient, but especially in the green up was especially found in the northern part of the gradient. And so, for example, this is senescence. So as things turn brown at the end of the growing season, that happened about a month later in the exotic sites and in the native sites. So these invasions are really affecting green up and senescence. And this effect is actually greater than the effects of temperature. So I think this is going on over time, these invasion by non-native species, and we kind of have to take that into account when we look at phenology. So here's the crown vetch again, November. It's really green when all the native species are dormant. So that was a big difference between native and non-native species. Okay, and in this Iowa climate assessment, I also looked at several other ways that the tallgrass prairie region is being impacted by climate change. And if you look at the tallgrass prairie, there's been 2 studies so far that have varied temperature, high temperature or ambient temperature. One was in Oklahoma and one was in Kansas, and one was by Phil Fay, who was in Temple for a while. Both of these studies found if you warm plots experimentally, forbs decline. So the proportion of grasses goes up and the proportion of forbs goes down. So forbs are really important to pollinators. And so the increased temperature and heat waves are probably going to cause a decline in pollinators, which is kind of concern. And one species of butterfly, the monarch butterfly, has been in the news a lot because of population declines, and it's going to be really negatively affected by climate change, because there was a study by Zylstra et al., and they analyzed all the data for this migratory population over time. And they found that climate variables explain 7 times more of the variation in their abundance than habitat. And so, like we're all talking about putting milkweeds out there and nectar plants, which I'm doing that too. But the climate variables were really important. And interestingly, the extreme years were really harmful for monarchs. So if you had really hot or really cold years, or really wet or really dry years, that led to declines in monarchs. And I think it makes sense because of this migration where they spend the winter in Mexico and go up through Texas and up to Iowa and Minnesota, and then they turn around and go back. That's such a subtle thing. They're used to kind of average years, and so extreme years are detrimental to monarch butterflies. Then, of course, another thing that's really important, and I live in Iowa now, and we don't have Johnson grass. And so there's just a little bit of Johnson grass in southeast Iowa. And this picture I took from one of our plots. And this plot used to be a 9-species mixture, and it was taken over by Johnson grass. And that bar is 1 meter tall, so it's really, really tall and aggressive. And if you look at the northern distributional limit of Johnson grass, it's limited by cold temperatures in the winter. So having a frozen ground for a long time period kind of prevents its northward spread. And so climate change is going to affect temperatures, especially the minimum temperatures. And so we're starting to worry about Johnson grass spreading up into Iowa. And we found it in Ames. There was one plant that showed up. We're in central Iowa, and somebody pulled it up. But these are the kinds of things that are going to start being surprises in the future, that things are going to move up from the north, I think. So our gradient analysis with Leanne Martin, we found that species diversity was lower in exotic sites than native sites. And I just wanted to talk a little bit about our experiment in Temple, where we kind of tried to look at the mechanism behind these species diversity differences. And on the right here, I have a plot, and these are sites in central Texas that we sampled. And this species diversity is on the y-axis, and the proportion of the vegetation that's native species is on the x-axis. And you can see they're related. So the more native species you have, the higher the diversity. And NBP means never been plowed, and PL means it was formerly plowed, and it's assembled into a new community. It's not a crop field, but it's a grassland on formerly plowed fields. And anyways, you can kind of ask the question, why are there these differences? And maybe it's just because they were plowed in the past, or maybe it's due to the species themselves and the interactions among species. And we wanted to test this, so we set up an experiment in about 2008, and we ran this experiment about 12 years. And I went back every year, twice a year, to sample it. And basically, the plots are set up so that they're either all exotic species, kind of like an invaded site, or all native species, kind of like a restored site. And then we had a summer irrigation treatment of about 5 inches of rainfall. So either plus 128 millimeter or 0. And we did this with lots of 64 mixture plots. and they were all set up so that they had 9 species to start. And we kept the functional groups constant. It was about 2/3 grass, 1/3 forbs. And then we had everything planted in monoculture, which just means a single species. And then we looked at a whole bunch of different things, microbial communities and fungal communities and plant diversity over time, and lots of different things that I'll just kind of summarize briefly here. But so here are all the species we used, and you're probably familiar with a lot of these names. So this is Temple, like Barbara's buttons, and was paired with dandelion. And then up the grasses, we had KR blue stem, and that was paired with little blue stem, and the coastal Bermuda with buffalo grass, and so on. So we drew 9 species that were exotic, and then the 9 species pairs, and then we planted a native plot. And so we did this right next to each other and compared them over time, and to try to really look at the mechanism behind what, any differences that we might see. So here's a picture of the Johnson grass again, and then a native plot real early on. So you can see it has lots of forbs and grasses, and a lot of the exotic plots ended up becoming really dominated by a few species. The top picture is us kind of in later years. So anyways, here's a summary of 12 years of work. So this was a ton of work and all just in one figure. But this is the number of species per plot over time. So you can see they're all planted with 9 species, and we let them go and we followed them for 12 years. And the green and yellow ones on the top are the native communities, and the red and black on the bottom are the exotic communities. So the native communities just maintain higher diversity over time, and they're really nice grass forb mixtures. If you stood out there and a butterfly flew over, they all went right to the native plots. And they didn't visit the exotics at all. Bees all went to the native plots. It was really dramatically different. And the irrigation treatments had really small effects compared to that difference. And what was interesting is the native plots responded to the summer irrigation, at least in some years, and the exotic plots didn't. So that was interesting. They did, the natives responded, the exotics didn't. And here's a picture of Pedram Danishkar, who worked on the project as well. And there were several exotics that showed really strong dominance. Klein grass, which I saw in Africa, love grass, Aerogrostis curvula, which is from South Africa, Johnson grass, Cara blue stem, coastal Bermuda. There was one plot that had crown vetch dominating for a little while. So it wasn't just a single species, it was a general thing. And the mechanism behind it was niche partitioning occurred in the native communities. In the exotic communities, it was completely missing. And what I mean by that is, in the native communities, things grew at different times of the growing season, and they had different rooting depths. So yeah, in the native communities, there were shallowly and deeply rooted species, things that grew in the spring and fall, and things that grew in the summer. And in the exotic communities, there is basically none of that. So the niche partitioning explained all of our results. So the plots are out there long enough, we could start looking at carbon changes in soil carbon stocks. And the soil type was the Houston black clay, which is a vertisol. So we had time 0 soil cores, and then we compared after 12 years how these native species and exotic species affected soil carbon. And what was really interesting is soil carbon after 12 years was higher under the native plants than it was under the exotic plants. And what was even more interesting to me is it only occurred at deep soil layers. So the 30 centimeters is about the first 12, about the first foot. And there was no difference in the first foot, but if you look below that, there was a lot more soil carbon under the native plants than under the exotic plants. And most people don't even look below 1 foot. They look at the surface only, and they're missing all of this carbon that's under native species. So these native species facilitated soil carbon storage. It was pretty interesting. We ran this for a while, and in the last couple of years, we did a seed addition experiment, and we did that to see if we could kind of obliterate all the differences that occurred. And so we split the plots in half, and half of the native plots got exotic seed mix, and half of the exotic plots got a native seed mix. And we did that to see if the differences would kind of disappear, and look at if could you restore the exotic plots with native species, and then the native plots, are they vulnerable to invasion? And I'm not going to show you too many details, but what we found was when you the mixtures basically resisted invasion, and the monocultures let everything in. And so we found that diversity is really important. If you want to resist invasion by exotic species, you should maintain high plant diversity. So the higher the diversity of the plots, the fewer exotics got established. And so I came up with this model for how Blackland Prairie is probably converted. So it starts off high diversity, then diversity is reduced for some reason, and then exotic species get in there. And then once they get in there, they have higher germination than native species, and it becomes more exotic dominated over time. So the question, I think, is why do these introduced non-native exotic species, do they differ from native species? Why do they have higher germination? And I tested this in a big greenhouse experiment, and I tested kind of two ideas behind that. One, maybe the exotic species were just selected to be kind of extreme genotypes. Like I read a book about the history of King's Ranch, and they said they went over to Asia to find grasses that were better than our North American species, and they introduced them on King Ranch. And I think when they did that, they probably didn't get an average species or a species that's hard to grow. They tended to get really common, kind of aggressive species. So, like I said, in Tanzania, I saw Klein grass, and it's actually very common in Tanzania, and it's very productive. And so that's the one they got from that area to bring in here. So maybe that's why they differ, or it could be they were produced when we brought them in. They produced cultivars. So it could be, okay, when they brought them in, they produced cultivars, and it's because they're cultivars that they differ. So I wanted to test that in an experiment. And what I did was I used the USDA germplasm, and I got seeds that were either cultivated genotypes or from their native range, non-cultivated. And then I compared those to the native species pairs that I used before. And then I seeded them, and then 21 days later, I added a 39 species prairie seed mix and let everything establish. And I did this in a greenhouse. I didn't want to put any of these seeds in nature, of course. So this was in a greenhouse. In the bottom left panel, the one on the right is little blue stem, and the pot on the left is KR blue stem. And this is after 21 days when the prairie seed mix was added. So you can see the difference. And then, of course, I had some bare ground plots that I added the seed mix, let it go a whole like 10 months, and had a lot of establishment. And it was really interesting. This was a fun study to do. Well, we used 14 species, so 14 exotics and 14 native species. And then the exotics were either wild germplasm from their native range or cultivated germplasm to test these ideas. And so here are the results after 10 months. So this is seedling germination. And we found that the exotics had higher seed germination than the natives. and it didn't matter whether they were cultivated or wild. So they were just consistently higher than the natives. And then this is the results from the seed additions from the 39 species seed mix added. It's kind of like a restoration study. And I'll just point out the top right panel is the number of species per pot after 10 months. And no priority meant that it was bare ground when we added it to the pots. And then the bar with that says native, a native species was seeded 21 days earlier. And of course, that's replicated 14 times. That's the average. You can see there's no difference. There's about 6 to 7 species per pot. And then over on the left, you can see exotic cultivated or exotic wild genotypes, and they're really reduced. So there was about 2 species per pot. In the bottom panel, Simpson's diversity in the dashed line is a monoculture. So if an exotic species was seeded before, it basically almost formed a monoculture. So to get back to the original idea, we ruled out that it was a cultivated versus a wild type. And basically, it did show that these exotic species are consistently different from the native species. They just have higher germination and higher growth rates. So I have about 8 minutes or 9 minutes or so, and I was going to talk about restoration ecology. And I wanted to point out, I wrote a paper a couple of years ago, and I just made a point in this paper that for the first 20 years of my career, I studied global change and climate change. And when you in that field, everybody's looking forward. So they're trying to predict the future. And about 20 years ago, I started studying restoration ecology. And I did a lot of restorations on my property and in different places. And I noticed restoration ecologists are always looking backwards. So they're looking back and they're going, okay, what communities did we have pre-European settlement? How do we bring those back? And it kind of makes sense, but the problem with that is we're not going to be able to bring back the climate of the past. And so I always kind of joke around, if you're going to do a real restoration, the first thing that you would do is lower CO2 to 270 parts per million, because that's what it used to be. Then you'd get started. And so obviously the dilemma is, how do we bring back the communities from the past when we can't bring back the climate of the past? And so that's sort of a dilemma for restoration ecologists to deal with. And there's lots of differences. And one is like the seed bank is really different. The seed bank used to be native species, but now it's native and all these non-native species that I just talked about. And this is a picture from Northwest Nebraska. And this is an exotic species called yellow sweet clover. And I went to this site because it was a remnant prairie. And if you think about doing a restoration here, you're going to add your native seeds on top of all of this yellow sweet clover seed. And so it's going to be really different than what we saw in the past. So it's a dilemma. And I've thought about what the answer to this dilemma is. And all I can really come up with is, I think if we restore biodiversity and get high functional diversity and restore the key processes and prairie systems, the right fire and the right grazing, then we might be able to get biodiverse systems back that can resist invasion and deal with all this variability and changing climate. And so just one example of this is a working group that my former student Forrest Isbell led, where he invited a bunch of us from all around the world who had done experiments where we set up plots with different numbers of species. And there's a little triangle for Temple there. And that's all of the work that we did in Temple. And then we used a couple data sets from Iowa. And then we joined with Europeans and others. And it was a big, giant meta-analysis that looked at ecosystem stability as a function of how many species were planted in the plots. And stability is producing biomass consistently. So if it gets really dry, it doesn't drop too far. And if it gets really wet, it doesn't go too high. And what we found in this meta-analysis is ecosystem stability and resistance, which is not changing when you go into a drought period or a flooding period. They were both higher with higher biodiversity. So I think if you want stability and resistance to all these climate changes in terms of their variability, I think biodiversity might be a way forward. It's probably going to take a lot more than that, but biodiversity will be part of it. So I've done a lot of work over the years on trying to understand biodiversity, why it's high and why it's low. So, but basically that's it. And just kind of in conclusion, I think elevated CO2 is having pretty large effects that are directly affecting plants and vegetation. And I think what we consistently find is tissue nitrogen declines, which should negatively affect grazing mammals and grazing insects and things like that. And then grassland invasions are sort of problematic. There's lots of non-native species that have been introduced over the years, and they're altering phenology, biodiversity, and soil carbon storage. And then a way forward is probably to restore more native species and more biodiverse sites. And when we do that, we need to stop looking backwards and try to look towards the future and maybe develop communities that are diverse that might be able to resist all of these changes in rainfall and temperature that are widely varying, I think. Not just the mean, but the variation. All right, and then finally, just arrival order is really important. I mentioned one of these priority effect experiments in the greenhouse, but we've done several more. And we usually find that when species arrive is important. So if you want to establish native species in a restoration, if you can get them out there early before the exotics arrive, they can really help prevent invasion when the exotics arrive. And I wouldn't wait on, I wouldn't try to get them established later with overseeding. They're kind of vulnerable early on. So anyways, I'd just like to acknowledge lots of people, especially Wayne Pauley, who I worked with for about 20 years in Temple. And anyways, I'll take questions, I guess.
Thank you, Dr. Wilsey. Does anybody want to ask a question about this? If so, you can just unmute and ask it or chat it and we'll convey it. I wanted to ask just a point of clarification, Dr. Wilsey. And when I see Trey, I'll read yours next. The first point you made about increasing CO2, and I just want to make sure I understood. So what you said was increasing CO2 means that the plants, that there's more biomass, plants will produce more biomass,
Yeah.
They'll get bigger, but they don't have as much protein.
Correct.
They don't have as much, basically, nutritional value.
Right, the percentage of protein goes down. But so if you're an herbivore and you're feeding on plants, you have more food, but then it's lower quality.
Mm-hmm.
And we see that over and over.
Mm-hmm.
And so there's been research for many decades on, how herbivores are going to deal with that. Do they just eat more? People were worried about insect outbreaks in forests. Like maybe they'll just defoliate trees. And they do that to a certain extent, but then they found that they're more vulnerable to predation and parasitoids because it takes them longer to develop. And then if, a lot of insects are limited by parasitoids, which lay eggs inside of the caterpillars. And then, so if they develop really slowly, they're more vulnerable to that.
Mm.
And then grazing mammals, if it's a large grazing mammal like a bison, they spend a lot of their day grazing. And they don't really have much more time to graze longer. And so it may just have a negative effect. And then of course people might counter that by fertilizing, because you can fertilize, but then there's a lot of problems with eutrophication. So we'll kind of see how it plays out, but
So it's not as simple as just putting nitrogen back.
right, correct.
Mhm. Yeah. Well, Troy Granger is asking, what level of biodiversity seems to be the threshold to stability and resistance?
Yeah, that in those studies, it was about between 4 and 10 species per square meter. Like, and especially if you get functional diversity, so you have things growing at different times of the year and different rooting depths. That's enough, it seems like, to have the effects.
We have a question, let's see, from Leslie asking, and based on what you know, particularly about Central Texas, I mean, what would be your top, the top of your list, a handful of species at the top of your list that you think needs to be in any restoration?
That's a good question. So, so, I like compass plant. So compass plant is a really, it's not used very much in restorations. It's more common in North Texas, but there's still quite a bit in Central Texas. And then a lot of the forbs, like Salvia azurea, I can't remember the common name, blue sage. And then we just looked at Mexican hat, which is, Orchibida colonifera. And I think blazing star is really good, Liatris.
Mhm.
So that's my favorite species, I think, in prairies. And up here we have like 3 or 4 species that are common. I think that's a really magnet for pollinators. And then I think a lot of our recent research, we've found that if having things flower early and really late is important. And so I think a lot of the asters, they flower really late. And like right now, all of the pollinators are visiting those.
Mhm.
And then early, we have past flower here, but I don't think that's in Central Texas. But I really like Barbara's buttons. I just think a lot of forbs, because I am partial to grasses. And so I think we just talk about the grasses a lot, but I think we forget about the forbs sometimes. And one thing I didn't mention is we found that forbs are really important in recovery from drought. So do you remember the 2011 drought? It was really bad in Temple. And our field actually looks like, I went there in October and it looked kind of like the field had been hayed, like it didn't really produce a canopy at all. And we followed those fields and those plots over time. And in 2012, they started to recover. And by 2013, they were recovered. And the ones with forbs really recovered, just right back to pre-drought conditions. And the ones without forbs, they were still way below the pre-drought conditions. And that's been found in other studies too. Some of the forbs are really deeply rooted, and I think they help prairies recover from drought.
Yeah, we find here, I don't know what anybody's experience is, but we're finding that forbs are harder to to restore basically than grass.
Yeah,
Grasses are much easier.
yeah, yep. They are hard.
Mm.
So we're doing a lot of work on, uh, how to establish them better and more easily. And
Um,
a lot of the recent work that we're doing is in Iowa, though. But in Iowa is different than
mm.
Central Texas. But But up here, forbs come in like crazy. It's sort of amazing. So,
And we have another question. In a restoration, I mean, do we have any control over when invasives arrive? It seems like, well, in our world, we start with an area full of invasives that we try to eliminate and then restore.
yeah,
And try to restore. I mean, is that a typical, what
yeah,
you,
yeah. Oh, and one thing I forgot to mention is we did a recent, which would be really helpful in Central Texas, is we did a recent study and we compared adding seeds to adding plants as transplants. And we used forbs that are hard to establish.
Hmm,
And they actually really established much better when you use transplants. And
yeah.
the transplants weren't that big. They were only like 3 or 4 weeks older than the seeds. So we just gave them a little bit of a head start, and then establishment was much higher.
Yeah.
And I think that's probably going to be helpful for the ones that are really fussy and hard to establish. But
Yeah. Yeah, I mean, it's worth the extra time
yeah,
and labor to do it
yeah.
that way. For
Of
sure.
course, some species it's not worth it. They're really easy probably, but
Yeah,
those fussy ones, I think that's probably gonna help.
it's gonna take.
Sure.
Here's a question. What would you determine to be the optimal grass forb ratio in a diverse prairie for the best soil and wildlife pollinators?
That's a good question. We're we're really studying that a lot lately, but the up here, and I think down there, I'm trying to remember what it is for Central Texas, but it's the native prairies that are really high quality are actually 48% forbs and 52% grasses. And so they're pretty forby. And so we have an experiment set up that we're in the 5th year now. Nathan Soley's worked on that, but we have 11 different levels, all going all the way from 100% grass, 0% forbs to 100% forbs to 0% grass and 10% increments. And we consistently find that it's better to have kind of roughly equal grasses and forbs, about 50% forbs, 50% grasses. And we found it's better for preventing invasion. It's better for pollinator visitation. It leads to greater establishment of plants. It's even forbs. And I think it's because they do a little bit better in establishing if there's a little bit of grass versus no grass. Then of course, if you have too much grass, it's really lower diversity than the 50-50.
Yeah.
So I think 50-50 would be a good target.
Mm.
In Iowa, it used to be forbs seed was more expensive than the grasses, but that's changed a lot. And so now they're not really any different. There's a lot of forbs that are really cheap. So I think it's good to get them out early and that's what I would shoot for. But
Yeah. What is your, here's another question from Trey Granger. What is your kind of total recipe in terms of, the elements you use, the tools you use, herbicide, fire, seed? What all do you do in restorations?
yeah. So we did some restorations when I was in Central Texas, and it was harder than here in Iowa. It's just hard to get things established from seed. That's why I was kind of leaning towards transplants because coastal Bermuda, pastures actually use plugs. They usually plant them as plugs. And so Central Texas, it was hard. And in Iowa, it's easier, but we tend to start with a crop field. And when they used to tell me that, oh, I would just grow a crop for a while. And that sounded crazy, but when I first got here, we tried to overseed exotic grasslands. Here we have a brome grass, Bromus inermis, which is our KR blue stem. And I used to think, oh, we'll just overseed it. And we would overseed it and nothing would come in. And so what they do really quickly is they've decided, you need to get rid of the brome or the caribou stem. So they usually use herbicide to kill it, or they even plant a crop. And
Mhm.
so like you plant like oats or something like that,
Mhm.
and then harvest the oats, and then you have an oat stubble, and then you seed on top of that. And I think in around here, I think we've found that it's really good to mow it the first year, like, because it takes a while for things to establish, and when they come in, they mostly grow roots. And so if you mow it high, you won't mow any of your seedlings. So mow it 4 to 4 inches the first year. And then we usually try to burn it the first fall. And then like here we get foxtails, like ceterias a lot. And then they come in the first year and then you burn that off. And then the second year is when you see a lot of establishment. And so here the big question is when should you add seeds? And everybody debates that. But I found that adding it at kind of at the end of the winter, like here we do it March 1st, the first week in March, because your seeds aren't out all winter to get eaten by animals, but then they still get some freeze thaw. So down there it might be a little bit earlier,
Yeah.
but I think that's important. And they've found over and over, if you seed in like too early, animals eat the seed in the winter. And I just always worry about that. But
Yeah. Yeah.
so that's what we do. And
Okay. Well, we just appreciate the fact that you have worked down here and kind of appreciate some of the challenges that are specific to
yeah,
here. We really appreciate your time tonight, Dr. Wilsey, and your expertise. And we wish you the best of luck with all of your research and your restorations.
thanks.
And thank you so much.
You're welcome. And good luck to your restorations too.
Thank you. We need it, don't we all? Good night, everybody. Thank you for being here.
Thank you, Brian.
You're welcome. Bye.
Night.